So you can see my screen?
Yes.
Yes, yes. This research is my research, and okay, I will just introduce it briefly. In my research, I studied the effects of cyanobacteria-derived organic matter on the estrogen biodegradation in the sediments in the, one eutrophic lake, Taihu, in China. And I found that the input of the cyanobacteria organic matter significantly increased the biodegradation of estrogen in sediments, which is mainly due to the stimulated bacterial activity, as indicated by the higher enzyme activity and increase the production of the extracellular polymeric substances. In the meantime, also the bacterial community composition was changed, and leading to the clear upregulation of metabolic function genes of these contaminants. I also used correlation-based network analysis and found that a strong facultative coordination between the community members and the compositional variability of the cyanobacterial organic matter in the co-metabolism. So our research suggests that the cyanobacteria blooms may be dominating the zones, may be the hotspot areas for estrogen activation. Maybe it's also for other emerging contaminants such as abiotics and pesticides and so on. So that's my research. If you have some questions. So if we don't have questions, then we see next one.
Hi, good morning. There is a mistake. I am the one who will expose Andrea Gonzalez. Can I start?
Yes, yes, you know.
Can I share my screen?
Yeah, you can.
Thank you.
You need to stop sharing your screen that Andrea can share her.
No me da el acceso. Ah.
Now you have access. Yes.
Thank you.
Remember to say only the main finds, right?
Yes.
So I think you can just introduce your work briefly in about 2 or 3 pages.
Can you see my screen?
Yeah.
Thank you. Hello, everyone. My name is Andrea Gonzalez, and I'm a master's student in the graduate program in marine sciences and limnology in Mexico. The title of my work is The Effect of Tetracycline on the Demographic Response of 3 Species of Rotifers. Currently, there are 7 billion inhabitants. Rapid growth of the population requires products, services that meet their needs, and then to pollute both aquatic and terrestrial environments. In the Valley of Mexico, 75% of the water bodies have some degree of contamination. Emerging pollutants are defined as compounds of different origin and chemical nature, such as cleaning and disinfection products, herbicides, pesticides, cyanobacteria, endocrine disruptors, and drugs. Their introduction into the environment is constant, which makes them a risk to public health. Tetracycline is a broad-spectrum antibiotic that exhibits activity against a wide range of gram-positive and gram-negative bacteria, organisms such as chlamydia, mycoplasma, rickettsia, and protozoan parasites. Tetracycline is one of the most widely consumed antibiotics worldwide, the second in Latin America, and in Mexico was the second most consumed until 2010 before the policy of non-prescription sales. Rotifers are used in toxicology because their population dynamics is well characterized and useful to investigate ecological and demographic principles. Organisms were isolated from the Cuemanco Canal and they were transported to the laboratory where a monoclonal culture was established. A main later concentration test was performed from a 200 milligram per liter stock solution. And there are the results of lethal median concentration. Here we can see that by increasing the concentrations of the antibiotic tetracycline in the medium, a significant decrease in population density and intrinsic rate of population growth was observed in the 3 rotifer species, the most visible effect being in Brachyonus caliciflorus. In the live eyeball, survival was reduced as the concentration of the drug in the environment increased, and in the case of fertility had a similar trend, since the reproductive period and the number of individuals produced decreased. The concentration tests here are the higher than those reported in natural water bodies. However, organisms in nature are constantly subject to the toxicants. Studies should be conducted to the test effects of the toxicants at very low concentrations, but over the numerous generations to simulate natural conditions and extrapolate laboratory findings. Thank you very much for your attention. have you the question or comments?
Hi, Andrea. Very nice presentation. I'm just curious about to know if you know that is there any kind of treatment to remove this kind of contaminants?
Can you repeat the question?
Yeah. Have you thought about any suggestion to remove this kind of contaminant from the water?
Yes. Yes. The wastewater treatment plants are not efficient to remove these contaminants, but I suggest that a regulation that indicates the límites permisibles, no sé cómo se dice, that indicates concentrations levels that not affected the water bodies.
Okay, thank you.
Thanks.
So we can.
There isn't any kind of treatment in these waters to remove it's basically the same questions of Christian, because I was curious. There isn't any kind of treatment, any kind of mitigation or nothing in these waters at the moment.
Yes, respect to other techniques.
Yes, other techniques. If
there are removed with UV light and ozone treatment, and the authors indicate that there is good, that antibiotic is removed with efficiency approximately, if I remember, that 70% approximately.
May I add a point over here? May I add a point?
Yes, of course, please.
Thank you. I just wanted to add that, yes, it may be possible to remove these nutrients by specific means, but the cost-effective ratio is very, very low, and therefore it is not done. And we just wait for natural ways to decompose these substances. Some of them may decompose fast, but some may decompose slowly. The other point is that since these are antibiotics, they are constantly being released into the system. So the processes to reduce these type of toxins, including cyanotoxins, is very, very expensive. And that is why it is not normally done, and hence the problem, especially when you refill many water bodies with partially treated wastewater. Thank you.
Thank you.
Thank you.
So we can ask the next one to just present your work briefly. Next is Alejandra.
Hi, it's me. Have to show my presentation, or maybe I do a little introduction like you did with yours?
Yeah. I think if you can the PowerPoint presentation is better. Yeah.
Yes, I have my PowerPoint, but I don't have a short version because I thought it was, the dynamic was different. I didn't know that I have to present it now, but I will. I can put my presentation and I can talk it briefly.
Yeah.
Okay. Well, the aim of my work is to evaluate 2 pesticides in Corbicula fluminia that for us is an invasive species, and to see if those 2 pesticides we decided to work makes DNA damage in the organisms. We choose 2 biomarkers, micronuclei and comet assay. What macronuclei are little DNA bodies in the cytoplasm that were broken at the end of the cell division. Here is in Spanish. Sorry, in the cell division, and comet and a comet assay shows the damage of the DNA with electrophoresis. So if you see the electrophoresis like a comet with the head and the tail, this means that your DNA has more damage. So we worked in the Laguna del Sauce lagoon, and we decided to work with 2 pesticides, metolachlor and metalaxyl, because metolachlor, that is here in blue, appears to be in all the sediments of the lagoon. And when we study pesticides in the muscles of fish, we saw that metalaxyl was present in all of the species, most of all of the species. So we worked with these 2 pesticides. So we exposed Corbicula fluminia at different concentrations in the lab with a positive control and a negative control. The negative control, we get the organisms from the field and take them to the laboratory with the control conditions, with temperature, light, and they were fed daily with phytoplankton. And for the positive control, we use peroxide, I don't know how to say it in English very well, hydrogen peroxide. This substance is known that makes DNA damage, so that we use it for positive control. After that, we exposed the organisms with different concentrations, and so we compare the damage with the organisms exposed with the 2 pesticides and the ones that were acclimatized in laboratory, and then we compare also with the organisms that were in field. So for metolachlor, we saw that also for micronuclei test, we saw difference with the negative control, with the lowest concentration. But when we saw the damage with the comet assay, we saw difference with the negative control with 0.1 micrograms per liter. And with metolachlor, we also see difference with the lower concentrations with the negative control, but for the other assay, we saw difference with a higher concentration. Afterwards, we compare the organisms of field versus the ones in laboratory with the control conditions. And we saw that the organisms in field also have more damage than the ones that were in laboratory, except for one place that we get the organisms, that was one effluent. We saw that in this case it was the only one that we didn't have difference between the negative control and the river study. So in conclusion, we say that Corbicula fluminea is a good organism to see evidence of damage for pesticides. So the aim of this work is to use these organisms as an early warning system to detect damage in organisms before it goes over to populations and communities and ecosystems. So we can take decisions in an early time before we see a big damage. Because in this lagoon, I didn't say it before, this lagoon is it takes water for drinking water for human. So that's the reason we want to look forward for assays or a worldly system to detect damage before we have a huge problem. That's very quickly. I'm sorry, my presentation was not in English, but in the I thought it was different. The thing. I don't know if someone has any question.
Hello, Alejandra. I would like to congratulations for your work. I would like two questions. One is about how animals were treated before extraction of hemolymph. And the second is about what do you think about apply this technique for another bivalves?
Okay. For your first question, we took hemolymph of the organism with a needle. So Corbicula shut the valves very, very hard. So I have to make a lot of pressure to pass the needle through the valves. So I have to break a little bit the shell with I have to break a little bit the shell. So after I take the hemolymph, the organisms were euthanized, were killed immediately with alcohol. But but also these techniques can be used with other bivalves. With Limnology fortunae, it's used a lot. And that organisms don't close the shell very hard, so you can pass the needle through the two valves and you don't have to break a little bit, and so you can use it for another occasion. Also, we are planning to use these techniques with native bivalve. So yes, you can use it for other organisms. Indeed, these two techniques, you can use it to any eukaryote cell. It doesn't matter if it's a fish, a bivalve, a human, you can use it with any organism. Or with any organism.
Thank you.
You're welcome.
Hi, Alejandra. I have a question. There are many kinds of organisms use a biocide test.
Yes.
So I would like to know if you found any advantage of using this mollusk in comparison with the other biotest organism.
Yes, we decided to use these organisms because they have a little low mobility. They are sessile, so you can take like a picture of that moment in that site. Also, they are fetal feeders, and these organisms have have pedal feeding, that they take the feed and consume organic matter from the sediment. So you have two matrices that you can catch the pesticides, because there are pesticides that get together with the sediments or get together with it gets in the water. You have different dynamics of sediment. So with these organisms, you can study two matrices, water and sediment, because they filter and they eat with the feed. Did you understand?
Thank you. Yeah.
You're welcome.
So if there are no question, let's ask the next one. Saritan?
Hello, yes.
Yes.
I prepared two slides to summarize my presentation, so I will be sharing with you. I don't know if you can see my screen.
Yes, we.
Okay. Okay, hello everyone. My name is Mayra Reyes. I'm from Mexico, and this is my first semester of the PhD. I am working on the effects of microplastics on the demography of freshwater microcrustaceans. As you know, the water ecosystems are simultaneously exposed to complex mixture of contaminants and to considerable variation of abiotic factors. The effect of microplastics have been more evident in the ocean by plastic islands or in the stomach content of birds and fishes by direct ingestion. However, in epicontinental waters, there's still no record of its presence, and the effects on the organisms with which it has contact. Cladocerans are generally filter feeders capable of consuming particles between 20 to 100 micrometers, which allows small particles of microplastics between 0.05 to 50 micrometers to be easily ingested, and this could have an adverse effect on their survival. The aim of this work is evaluate the acute effect of 5 different concentrations of microplastics, 2.5, 5, 10, 20, and 40 milligrams per liter on the 3 different life stages of Daphnia pulex, neonates, juveniles, and adults under 20 and 25 centigrade. We found that the organisms at any growth stage is able to consume, okay. Sorry. Yes. We found that any growth stage is able to consume microplastics and store them. The consumption of microplastics and its adverse effect was greater at 25 than 20 centigrades, and the adults show a higher sensitivity to microplastics in compared with juveniles or neonates. And that's all. Thanks.
So I have a question. In your research, the microplastics are dispersed in the water, or they coagulated?
For this test, I bought the microplastics, but in the medium, in the ecosystems, there are the presence of microplastics in lakes, for example. Mm-hmm.
Yeah, I don't know. I mean, you can study the effect of the organic matter on the on the effect of macroplastics on the aquatic organisms, because the interaction between macroplastics and organic matter is very common in waters, and the size of macroplastics will be changed if they interact with the organic matter, dissolved organic matter. Yeah.
Okay, okay. Yes.
Yeah. Thank you.
Thanks.
Hello. I have a question. Did you count MPs in the guts of Daphnia species?
Sorry, can you repeat the question, please?
Did you count microplastics in the guts of the Daphnia species?
No, no, I only measured the number of the organisms that present the percentage of the mortality of the organisms, but I don't count the filter of the organisms.
Okay, thank you.
You use a microplastic of 30 microns, right? Did you check the selective size of neonates of Daphnia? Because I think that the toxicity could be related to the size that they can eat. So I don't know if you check the size of particles that can ingest the neonates.
Yes, this is why we select this size of microplastics, and in the neonates also we saw the ingestion of the particles. So, but the effect is less because they don't ingest a lot of in compared with the adults. But yes, they have microplastics in the gut.
So, any question?
Yes, I have a question. Hi. Mayra, do you think that if you used aged, I'm going to write it in the chat because I don't know how to pronounce it. Microplastics could have another effect on your Daphnias?
Can you repeat the question, please?
Do you think that using those microplastics could have another effect on your Daphnias, on the mortality of your Daphnias?
Both microplastics?
Ajá. In Spanish it is como usadas.
Okay, okay. Yes. Some studies shows another effects, but the effect is associated with the decomposition or the degradate of other compounds that it's used to make the microplastics, like colorants and I don't know, the other chemics that make the microplastic or the plastics durability, or yes. So the effect can be about other compounds into the microplastic, not physically.
Thanks.
So if no question, let's ask the Jules and Antonio. Yeah, his research is also about the microplastics. So please. Jules, Antonio.
Okay.
Yeah.
Okay.
You can share your presentation and introduce your work briefly. Yeah.
Yes. So Can you see it now?
Yeah. Okay.
It's okay.
Okay. Well, I am Jose Antonio. I'm going to talk to you briefly about my presentation that has a title, Filtration and Ingestion Rate in Platinum Spatulus Exposed to Cadmium and Microplastics. Well, plastic production has increased since 1960 exponentially worldwide, and few strategies have been taken to manage this pollutant that can be transported by wind and water currents, reaching aquatic systems where it accumulates. In aquatic environment, plastics can be found as microplastics. These small-sized particles are capable to absorb other contaminants, such as heavy metals like cadmium. This heavy metal is used in the industry to produce alloys, batteries, and plastics. And despite the effort to investigate the effect of cadmium and microplastics separately, these studies have been conducted on the effect of microplastics alone or with other contaminants such as cadmium on freshwater zooplankton, particularly on freshwater rotifers. The phylum Rotifera has about 2,000 species, and they are the trophic link in aquatic systems. These small-sized primary consumers have a particular feeding behavior as they use the cilia of the corona to move and produce microcurrents for food intake. In the few studies available on marine rotifers, the impact of microplastics in life history traits is demonstrated. However, microplastics does not have any effect on the survival in acute toxicity tests. Therefore, the question is, do microplastics alter energy intake, and that is why they only show effects in chronic tests? As COVID-19 pandemic has changed our ways to do experiments, one way to evaluate rapidly a possible change in life history traits is to evaluate the feeding rates of Platinum spatulus, because this is the beginning of the process where energy is obtained for survival and reproduction. So, a diminution in this activity due to some pollutants can result in changes of the life history traits. This is Platinum spatulus, the species that was used in our investigation. This is a herbivorous rotifer commonly found in Mexican shallow lakes and in North America. It has been shown that it is sensitive to diets, pollutants, and different food densities, changing its demography. It is also capable for accumulating contaminants, and it has an effect on higher trophic levels, such as like in a predatory rotifer that is Axpelagna. Because of these characteristics, it has been proposed as a standard biocell organism. And well, the aim of the investigation was to evaluate the filtration and ingestion rates in Platinum spatulus exposed to cadmium and microplastics. These are the microplastics that were used in the investigation. And here are the results. The two figures shown above are the effect of cadmium alone in the ingestion and filtration rates of Platinum spatulus. And contrary to what was hypothesized, as the concentration of cadmium increased, the filtration and ingestion rates of Platinum spatulus increased. This can be explained by the effect hormesis, that it is a kind of stimulation to overcompensate the stress. In the first instance, this appears to be beneficial for the organism, but if it is not the only pollutant in the system, what happens when cadmium interacts with microplastics? Even if there is some temporary gain from hormesis, research shows that benefits have adverse effects on the long-term exposure. And then the other two figures that I show you is the effect of microplastics alone, or clean microplastics, and the polluted microplastics with cadmium, the sublethal concentration of cadmium. And here we can see a pattern. As the concentration of microplastics increases, the filtration and ingestion rates of Platinum spatulus decreases. And well, the increased filtration rates with low cadmium concentration did not change the fact that microplastics interfere mechanically with the food intake. The trade-off was that low cadmium concentration could have a beneficial effect on the fitness of Platinum spatulus. But on the contrary, when these heavy metal and microplastics interact, more energy is needed to filter and feed because microplastics interfere mechanically. The fact that some pollutants can lead to an increase and other interfere in food intake, it can lead to the formation of the corona and malfunctioning of its cilia. This put rotifers at risk since they are neonates, and considering their ecological importance, there could be changes in the community structure of shallow lakes that are more exposed to these pollutants as they are near to metropolitan areas. And well, the conclusions were that the pollutants tested had an effect on the feeding and filtration rates of Platinum spatulus. Microplastics alone decreased the ingestion and the ingestion rates of the rotifer, but sublethal concentration of cadmium increased this behavior. When the pollutants are combined, The adverse effect increased significantly when the concentration of microplastics was higher as compared only microplastics or clean microplastics. Despite the ecological importance of rotifers, which completely depend on filtering the environment to obtain their food, there is still no regulation of this emerging pollutant. An important fact of the investigation is that the concentrations of microplastics that were used, they are environmentally relevant. So urgent regulatory steps are needed to prevent undesirable changes occurring in the structure of zooplankton community in the shallow lakes. And that's all. Thanks.
May I ask a question?
Yes, please.
Yeah, particle concentrations and size have a very important effect on filtration rates of rotifers. In your treatment, you have different concentration of particles. Yeah, independently of the kind of particles, they affect filtration rates. Did you consider this part?
Yes, I consider this part. We use different concentration from low to high to see if there was an effect, considering that the size and the origin of the particles used.
Yeah, but it be an effect of number of particles, not of plastics itself.
Oh yes, well, all the literature shows that microplastics and some how do you say it, litter or clay only interfere mechanically, and also they cannot absorb these microplastics, these other contaminants. So there can be a synergetic effect. So this was also another thing that I tried to demonstrate. But to do this, I need long-term exposures and long-term experiments. So they are going to be the other step of the investigation.
Okay, thank you.
So if no question, let's ask the final one. Christine approach. Yeah. Okay.
May I share my screen?
Yeah.
Okay. Can you see it? Okay. Good morning, everybody. I'm going to present the study Morphological Alteration in Keratella from Lake Pátzcuaro, Michoacán. This study was done by undergraduate student Diana Martinez in a vacation stay in the summer of 2019 in a collaboration with the Mexican Institute of Water Technology. Well, Lake Pátzcuaro is the third largest lake in Mexico. It is eutrophic, hypertrophic, depending on the season of the year. It has an area of about 100 square kilometers and a mean depth of less than 5 meters. The zooplankton community in this ecosystem is dominated by rotifers, especially those of the brachionid family, mainly Keratella cochlearis, Keratella americana, Brachyunus caudatus, and Keratella tropica. This study is part of a program, a monitoring program, did between 2017 and 2018 at 13 sampling stations, as you can see in the map. So at the end of the dry season, we found just in the Embarcadero area at the south part that a couple of individuals of Keratella cochlearis had an abnormal pattern on its posterior spine. So we decided to go deeper analyzing this sample and to look for the morphological alteration of this species. So we number the spines, as you can see in the top left of your screen, as number 1, 2, 3, 4, and 5. And we check 12,000 individuals to look for any abnormality on their spines. So we only found 12 organisms, and these are the picture of these abnormalities at the posterior spine. And we show in this graph if one of these organisms have another abnormality in another spines. So we found that all of them present this posterior spine twist, and some of them present other kind of abnormality in other spines, especially in the spine number 2 and 3, as you can see in this. Let me show my pointer. Okay, well, this one. And we also found that Keratella americana, only 3 individuals show also a twist posterior spine. After this, we check for the egg-female ratio, and we also found that Keratella cochlearis, that is one of the most abundant species, had the lowest egg-female ratio. We also compare regular organisms against organisms with abnormality. We didn't find any organism with this kind, with egg, I'm sorry. We check in the literature about any similar tendency about modification of the structure of rotifers. And we found in laboratory experiments that sulfur compounds, pesticides, hormones, and heavy metal can cause morphological alteration, but none of these, as you can see in the center and in the left, are similar to those that we found. In a personal communication with Claudia Thaus, she commented us at the Rotifera meeting 2 years ago that she also found something similar in the Indiana state in the United States, but she neither know what is the reason for this abnormality. We suppose that this related to pollution, but we don't have any more data about it. Sadly, we don't have data for molecular analysis. It wasn't our aim in this study, but there is a very good opportunity for future research on this topic. And finally, this study is already published as a short communication in a local journal, and you can get it if you write me an email if you are interested, or you can also find it in the research guide. So the organisms found with malformation presented a higher frequency of morphological alteration in their posterior spine and didn't present any egg. So it could be deduced that the malformation are affecting the reproduction, but we need more data. Monitoring of the contaminants in this area of the lake, it's better if we do it in the whole lake, but this is highly recommended, and also some experimentation in order to verify the factors causing these malformations. Yeah, thank you.
Hi, Christian.
Hi.
Congratulations to your work. It's beautiful. I have a question. I don't know if it's silly, but in your work, did you measure the pollutants, contaminants, something like that, to say what is the reason for this malformation? So yeah, I didn't understand well.
Yeah, there are many kind of pollutants there. We measure the standard variables, turbidity, temperature, pH, oxygen, total and fecal coliforms, turbidity, oil and fats, and we found the low pH out of regulations, high number of total and fecal coliforms, high turbidity, high presence of cyanobacteria. It's an area with a lot of ship, of motorboats. But our aim wasn't to test this kind. No? We just found it passing by, trying to work with bioindicators. But now we want to return there to analyze what kind of contaminants are and high concentration, and maybe isolate this keratella, take samples for molecular analysis, and make some experimentation in the lab. We just suppose that could affect, but we need to test it.
Yeah, thank you. Because I was curious and I was thinking about hormones, because I know there is some research from hormones in the water that changes fishes, I don't know in English, but changes
chemical communication.
Reproductive
success
organs of fishes. So maybe if there is something about that for zooplankton, I don't know.
Yeah, we discard predation effect. We check the papers about plasticity, morphological plasticity of this species, and we didn't find any related to overfindings. So this is new, and it may deserve longer research about. Yeah. Mariana, you are muted.
No, I wasn't talking.
Okay.
I was thinking.
Okay.
Anyone has any questions about any work? Because we have time.
I have a quick question for Christian. Christian, did you see this malformation in any other species of rotifer? Or any other species of keratella?
Yeah, in Keratella americana, we found 3 individuals. Maybe if we look deeper in the sample, the whole sample, we may found maybe another species or higher frequency of these abnormalities.
Was there any other keratella in your sample?
Keratella americana, yeah.
No, but no other species?
I'm sorry?
In Keratella tropica, there was no change?
No, and it was the highest abundant species. Yeah.
Okay, thank you.
There are some papers at lab conditions, and they refer malformation in Brachyonus caliciflorus, in Philodina rosea, and there's a work of Coelho in 2019 who found concavities in Testudinella patina or marginula, but did discard the effect of contaminants, and they think that it could also be related to a recently hatched, you know, they have a slightly different form, yeah.
I think Mariana's point is interesting. The hormones, because hormones are plenty in this water body because it is, you know, a lot of sewage waste enters over there. So that may be an interesting, very interesting thing to look at. Thank you. Thank you very much.
Yeah, thank you to your questions too.
We still have time for questions.
Or for moving to another room?
Yeah, maybe. I have a reminder to say, today at 12 o'clock, we have the ceremony, the closing ceremony. So everyone is invited. Here in the chat, I put the link to the closing ceremony. So I hope we still meet there today. So this is it. No one has any questions? So maybe we can move to another room.
Okay,
thanks very much.
Have a nice day.
Thanks. Bye-bye.
Thank you.
Now I'm going to close the room. Thank you.